Good evening. On behalf of the National Stem Cell Foundation of Australia, welcome to Future Medicine. Can stem cells save sight? My name is Tanya Ha. I'm Director of Engagement at Science in Public, where I've looked after communication for the National Stem Cell Foundation of Australia for more than a decade, and I'm delighted to be your host this evening. I want to start by acknowledging the traditional owners of the lands from which we're each joining this event. I'm coming to you from the lands of the Wurundjeri people of the Kulin Nation. I pay my respects to their elders past and present and recognise any people from other First Nations who might be joining us tonight. Humans, we are highly visual creatures, and eyesight is a wonderful thing. It allows us to enjoy scenic landscapes, to find our favourite foods on supermarket shelves, and recognise the people that we love, or maybe the people we want to avoid. Goats have rectangular eyes, which is good for peripheral vision. An ostrich's eyes are bigger than its brain. And a chameleon has two eyes and they are independent. They can look in two different directions at once. But our human eyes allow us greater ability to read, work, drive, take part in hobbies and other activities from painting to playing darts. But eyes are also vulnerable to injury and aging. In fact, nearly 500,000 Australians are blind or vision impaired, which can affect their independence, work, and social interactions. Prescription glasses and contact lenses, such as the ones I'm fortunate to wear myself, they can help people, but some eye injuries and diseases are currently beyond repair or cure. The good news is that scientists and clinicians are working with stem cells to study and potentially treat different eye conditions. The National Stem Cell Foundation of Australia is a registered health promotion charity with a mission to promote the study and use of stem cells in the prevention or control of diseases to alleviate human suffering and to enhance public education in this field. Tonight, we're going to hear from three top scientists paving the way for new therapies and a better understanding of the conditions that affect our eyes and eyesight. They are Professor Stephanie Watson, OAM, a clinician scientist at the University of Sydney's Save Sight Institute, Sydney Eye Hospital, Sydney Children's Hospital, and Prince of Wales Hospital, who researches stem cell treatments for corneal damage and limbal stem cell deficiency. Associate Professor Anay Gonzalez Cordero from the Children's Medical Research Institute in Sydney, where she leads their stem cell and organoid research facility and researches stem cell therapy for retinal disease. And Dr. Tom Edwards, also a clinician scientist from the Centre for Eye Research Australia in Melbourne, who is one of a handful of surgeons in Australia who has expertise in retinal gene therapy surgery. I'll be talking to them more about their research and what we can reasonably hope for in the future. Later, you will have a chance to ask your own questions. Please write them in the Zoom chat box and we will get to as many as we can. Now, behind the Science in Public participant, the logo that you see is my colleague Jackie. She's providing technical support. You can send technical issues to her directly via the chat function. Also note that tonight's event is being recorded so that we can share it online at a later date. It now gives me great pleasure to introduce our first panellist, Stephanie Watson. Stephanie is an eye surgeon specialising in cataract, corneal and laser surgery with expertise in eye disease in adults and children. She's known for her translational research into stem cell treatment for corneal blindness, which has been supported by the Medical Research Future Fund. Welcome, Stephanie, and tell us what are your current roles and how do you divide your time between research in the lab and treating patients?
Thank you, and thank you for the kind introduction. I'm very lucky in that I have clinical roles and I also have research roles. And this enables me to really find out, what the patient needs are and then try to address them through my research. So at the moment, I'm head of ophthalmology and head of the corneal research group at the University of Sydney. I'm head of the corneal unit at the Sydney Eye Hospital. I'm also chair of Australian Vision Research and chair of New South Wales RANSCO.
Great. Tell us, I want you to give us a bit of an introduction to the eye. Tell us about eyesight and the human eye itself, and does it have the ability to repair itself?
Sure. It might be good to show the slide of what a human eye looks like for those that aren't familiar. But basically, the eye is like a camera in that we have focusing elements at the front, and at the back, there's a very sensitive tissue, the retina, which then sends the visual signal to the brain. Now, the eye does have some capacity for repair. An example of that is in the corneal epithelium, the very outermost layer of the eye. This layer protects the eye from infection and other diseases. And every 7 to 10 days, it's thought this layer can regenerate itself through stem cells. Here you can see an image of the eye and its numerous parts. Thank you.
Great, thanks, Stephanie. Is the eye special compared with any other part of the body? What makes it different?
Well, as an ophthalmologist and eye researcher, I like to think it's very special, but it's also, I know, very special to patients because patients would much rather, lose an arm or a leg than their eyesight. But it's a special organ in that it's composed of extremely delicate tissues and a variety of tissues. There's also a degree of immune privilege, and we can actually access it for stem cell treatments. We can also visualize different parts of the eye and its functions. And so it's an ideal model for developing stem cell therapies.
Great. Thanks for that introduction, and we'll hear more from you and ask you more about your research a bit later. It now gives me great pleasure to introduce our second speaker. Tom Edwards is an eye surgeon and leads a research team investigating retinal gene therapy at the University of Melbourne affiliated Centre for Eye Research Australia, based at the Royal Victorian Eye and Ear Hospital. In 2020, the foundation provided $50,000 to support Tom's research into gene therapies for inherited eye diseases through our match funding program. Good evening, Tom. Tell us about what your research is currently focused on.
Good evening, Tanya, and good evening, everyone, and thanks for joining us. Yeah, well, like Stephanie, I've got the fortunate position to both treat patients clinically and also lead a research team, and there is some overlap between that. For example, some patients we see in the clinic have genetic disorders that we then refer to our research team, and we're able to sometimes generate stem cells from those patients, which we can use then to make cells in the laboratory, which we can test new treatments and gene therapies on. And that was a big part of the grant that I was fortunate enough to receive from Stem Cell Foundation a few years back. So yeah, we're investigating broadly the group of disorders called inherited retinal diseases. Together, they're actually the commonest cause of severe vision impairment in working-aged Australians. And the tough thing about them is, by and large, they're incurable and lead to severe visual impairment in many individuals. And so our team are working hard at developing novel treatments and also facilitating clinical trials from exciting teams from around the world who want to come and do their research with our team here in Melbourne.
You were mentioning before that, inherited retinal diseases is your particular field of interest, or one of them. But can you tell us very broadly what are the common eye problems that Australians, more broadly around the world also, that people have, and what can we currently do about them?
Yeah, well, a good way to think about it is firstly based on the patient's age. So I mentioned before working age Australians, young Australians, the big disease burden there from an ocular point of view are these inherited retinal degenerations. And then at the other end of the spectrum, sort of the older cohort, you've got diseases like glaucoma and age-related macular degeneration. And then sort of overlapping both young and old is, of course, diabetes, which can be diabolical for eye health, and then in some young people, trauma as well can be an issue. So there's a large range of things that can go wrong with the eye, and it's our job to try and come up with ways to prevent that from happening, and if prevention is not possible, to come up with treatments and cures.
It's an interesting nature versus nurture kind of perspective, isn't it? Because some of it is genetic, some of it is in the genes and inherited, but some of it is the life we
Mm.
live, the environment we're in too. Is that correct?
Yeah, that's exactly right. And there is a bit of overlap. So in its simplest form, the inherited retinal diseases are purely genetic and usually caused by single genes, spelling mistakes on single genes. And
Mm.
so they're relatively amenable to a gene therapy approach where you might, for example, want to introduce a working copy of a gene into the affected cell to correct that error. But then you move into other more complex genetic conditions, well, at least conditions with a genetic basis that is more complicated, like macular degeneration or glaucoma, which do have a genetic basis. And you are at a higher risk if you've got an affected relative. But the genes is not the only story, of course. And then moving into other, well, obviously trauma. There's no that's entirely environmental. And diabetes, often also lifestyle related, but also genetic too. So you're right.
Mm.
There is a mix.
Good overview. Thank you, Tom. I'd now like to introduce our third speaker or panelist. Ana Gonzalez Cordero is a leader in the field of making stem cells from adult cells and turning stem cells into complex and functional 3D clusters of cells called organoids that recreate eye tissues in the lab. She is based at the Children's Medical Research Institute in Sydney. And in 2022, she won the Metcalfe Prize for Stem Cell Research from the Stem Cell Foundation. A delightful winner. What's your current research focused on, Anai?
Hello, everyone. Good evening. I hope you can hear me properly. If not,
All
please
good.
let me know. So I lead a research group. So I'm an academic. I do research. I'm not a clinician. So my passion goes entirely to research, and it's researching stem cells and the eye. And so I lead a research group at the Children's Medical Research Institute. I also have an appointment with the University of Sydney. I am the vice president for the Australasian Society of Stem Cell Research. I am a board of directors for Retina Australia. So I do try and engage as much as I can with patients and the research.
Oh, you're a legend in the stem cell field. Speaking of which, I thought I would give you the role of telling us, for those who don't know, what are the basics? What are stem cells?
Yeah, okay. So I'm sure you have heard about stem cells, and they are used very much on a broad term because we have different types of stem cells. So I'm going to try and explain very simply what they are. But I think I always like to do the analogy of stem cells being these very magical, special cells that are like babies. They are baby cells that can grow indefinitely. So they are like, they just proliferate and grow in the body where they come from originally in the very young embryo and in the dish because we can actually grow these stem cells in the dish. So these baby-like cells can go on to give rise, so to generate any other cell type of the body. That's why I say that they're like babies. They can choose to grow up to be a heart cell, a brain cell, an eye cell, a muscle. And the beauty is that if we're growing it in the dish, we know how to make them go into the direction, into the organ, the cell type that we want. Now, we have another type of stem cell that are more like teenagers, let's say. So they're more like they are set on what they will be. So they have a bit more personality because they live on that environment. Like if, as Stephanie mentioned, in the eye, in the cornea, we have a population of stem cells that they live in the cornea. So they still proliferate, so they grow, but they will only become that cell type of the cornea. So they're a bit more limited in what they can do. So that's the main difference of stem cells. I think
So it's like they've started to
summarizing.
narrow down their possibilities a bit and
Yes.
specialize a
So,
bit more.
and we
Mm.
find stem cells in the lung, in the gut. So various organs have these stem cells that are a bit more limited in capacity.
Yeah, yeah. Good description. And so how and why do you make healthy and diseased eye cells from patient samples. I've gathered that that's something that you can do, that you can take a blood biopsy and use those adult cells from patients to create stem cells, either that are healthy or that express the disease. Tell us more about that.
Yeah, so as I explained before, you in the past we used to get stem cells to grow in the dish from the very young embryo. So just after fertilization, there were that that population of stem cells exists. Nowadays, we are very fortunate that a very clever scientist back in and it's a Japanese scientist, Shinya Yamanaka. He won the Nobel Prize for medicine for this discovery. He discovered that you could get a cell that knows what it is, a blood or a skin cell, and deliver, so give these cells, turn on these cells for genes that are present on stem cells, on those baby stem cells. So he basically turned on those genes in the dish in vitro from the blood and the skin. And that by turning on those four genes, he was able to turn it back in time. So the skin turned back in time to become a stem cell. So now we
So,
went
like
from an
embryonic stem
adult
cells?
cell.
Yeah.
Sorry.
So it became like embryonic stem cells without actually having to use an embryonic stem cell line?
Exactly. So we went from an adult cell, so your skin sample, all the way back in time to embryonic like stem cells. And to be honest, if you look down the microscope, you cannot tell the difference between an embryonic stem
Wow.
cell and now this cell that has been generated from the adult. And that is very important and powerful because now we can generate these stem cells from every patient, every individual. And the beauty of stem cells, as I explained before, is that they can grow, right? We can culture them in the dish because they keep growing. And what we can do as well is freeze these cells. So we know how to freeze them in so that we can use at any time import in our research. So we can generate cells from the patients and from healthy individuals, grow them in the lab, and then freeze to use for research.
So you could take a blood sample from me, for example, and turn that back into stem cells. And then if I was showing signs of different eye problems, you could use the blood sample, turn back into stem cells to produce eye cells that have whatever genetics I have. And that's a way effectively of getting an eye cell biopsy without getting it from my eye. Is that right?
Yes, exactly. So if we go now a step further, so I have the stem cells, these cells that can generate anything, right? Any cell type of your body. What we've learned in the lab and my specialty is that we know how to make this stem cell into an eye cell, and more specifically into a retina cell, right? Any of the cell types of that we have in the retina. So it's what Stephanie explained, that tissue at the back of your eye where you have, for example, your light sensing cells, the cells that are responsible for vision, the photoreceptor
Ah,
cells. So
yeah.
we can now generate cell types, and I'm going to explain to you later that we actually generate these in 3 dimension as mini organs, and that has made all the difference in the field.
Yes. It's very exciting. Yeah, and I know that if I was a patient, I know how I would rather deliver eye cells to you as a biopsy from my blood veins, not, I don't fancy the idea of the injection going into my eye, but I know that's a reality for a lot of people. Okay, I'm now going to invite our other two speakers back for some general discussion as we dig deeper into this theme of can stem cells help save sight? I wanted to start by exploring this idea that, we've heard of stem cell therapies and they've had a lot of promise. They've actually been in use for a number of decades. Tom, I know that therapies like bone marrow and cord blood transplants, they've been around for literally decades to treat things like leukemia. Why aren't there more current stem cell treatments for other parts of the body, such as the eye, that you can use in your practice?
Well, I suppose it's important to think about, what you're really describing when you say stem cell therapy, because you don't really want stem cells in the eye. What you might want, however, are cells that have been made from stem cells, like retinal cells, to replace those cells that are damaged or lost in the eye. So that is a reasonable strategy to pursue, and people have been trying very hard. The difficulty is the eye is a very complicated organ, and the retina even more so. And it's one thing to say, well, let's put a light sensitive cell in the retina, but it's no good on its own, even if it were to detect light, because its other key job is not just to detect light, but is to pass on that signal to the surrounding cells in the correct manner, and to avoid uh, causing inflammatory or scar-like responses that might be mounted in response to its presence there. So it's really the complex anatomy and structure and delicate connections between retinal cells which have limited thus far, stem cell derived uh, cells being used. That said, there are other layers in the back of the eye that are perhaps more simple. There's a layer underneath the retina that is a perhaps simpler layer, and that perhaps could be the most promising cell type to replace, and will perhaps be the first one. And that would have promise for some diseases like macular dystrophies and macular degeneration. So yeah, there's a lot of work being done. We'll get there eventually, but it's going to take time.
Yes, and I guess it's early days, and we want to have enough of the right sort in the right place and not have too much. So, Stephanie, I'll turn to you. That we've heard before from an eye about stem cells in the developing human, embryonic stem cells, but also there are still stem cells residing in adult organs. Are there still stem cells in a healthy adult human eye, and how do they help?
Sure. Well, I can give you the example of what we call the limbal epithelial stem cells. They're the stem cells of the cornea, and they actually reside between the clear cornea and the conjunctiva in a specialized zone called the limbus. And they're in these sort of folded patterns to protect them from UV and any other damage. And these cells are constantly, producing new epithelial cells in homeostasis, just steady state maintenance, but also in diseases, they have big roles in repair.
Okay. We've looked, coming to you and that we've talked a bit about the potential like the broad potential that we had of stem cells to create replacement tissues. And Tom talked about that. And then Stephanie talking about the existing adult stem cells in the eye. I think that when there was a lot of hype and hope around the potential for stem cell medicine, regenerative medicine, the idea was that this was going to be a source of cells, tissues for replacement organs. But in fact, the history of stem cell science is that it actually ended up being a really powerful and useful research tool, not just potentially a treatment. Why do we need that? Can you tell us more about how stem cells are a research tool as well?
Yeah, yeah. So we use it for therapies and replacement therapies, and we're going to chat a little bit about that. But I think the beauty here and what you need to remember is that we're generating stem cells from a patient or a healthy individual. So, and then making them into mini organs, basically, into the retinal cell types. And we are talking about human cells, right? Traditionally, researchers like me, I studied zebrafish, I studied frogs. So, and it was fantastic. These models are very informative, and we learned a lot. Everything pretty much that we know of how the eye develops actually comes from these animal models, but they don't replicate. Sometimes they don't replicate very well the anatomy of the human eye, how the human eye behaves. So having that model, that those retinal cells that are generated from the human stem cells, it's very powerful for us. So now, if you imagine we generate a stem cell from the patient, from the blood, so we have the blood as the initial sample, then we make the stem cells, then those stem cells are made into the retinal cell types, And those retinal cell types, the DNA, so the genes in all these cells, all the way from the blood to the retinal cell types, is the same. So the retinal
Mm.
cells that we're now growing from the stem cells have the disease or the mutation that the patient has. And that
Mm.
means that if we grow the retina, now we perhaps are able to model the disease, to grow the disease, the organ with the disease in the dish. And
So
we
does
can
that
grow thousands and thousands of these cells.
does that mean that you can look at how these tissues function with the influence of those genes at that cellular level and maybe even that molecular level to see
Mhm.
how the what's the mechanisms of the diseases? How do they work?
Exactly. So now we have in the dish patient-specific retinas growing that we can because as it's we work in genetic conditions of the retina and they're very heterogeneous, so they vary a lot, and it's important for us to understand why are they varied, what is that gene, the defect on that gene causing to the retinal cell types. We understand what the gene, the defective gene is, but we don't necessarily understand the whole pathway that will lead to that cell to perish and eventually die. So
Mm.
having those models, it's great. We can, yeah, understand disease and
Yeah.
after that then test new therapies on them.
Yeah, and I imagine that it would give you some clues to what the potential treatments might be or what's existing among our library of existing drugs and treatments you could use. Tom, I'll turn to you. What's been your experience of using stem cells, not just in the clinic, but in the lab?
Well, in the clinic, I could start with because it leads on nicely from what Anne was talking about just now. In the same way as for research, stem cells can be made into retinal cells from an affected individual so that we can learn more about the mechanism, the genetics, the molecular biology, etc. You can do that exact same thing in a clinical setting when you're trying to confirm whether or not a patient has a particular genetic condition. Um, and what I mean by that is when you do genetic testing, and some of the audience may have been through this, you're generally counseling a patient that, well, it might be clear cut. We might find a gene that is responsible for your condition, but there's a reasonable chance, perhaps something like 30% of the time, it's not clear cut. And that group, sometimes you find a change in a gene and you're not sure whether it's significant or not. It might be the cause of the problem, but it might just be a natural variant. And that's just what, we all have these differences. That's what makes us all a bit different. And so we had a pair of patients, they were related actually, they were cousins. And at least from the clinical perspective, that their retina looked very much like they had an inherited retinal disease caused by a change in a gene for which there is now a treatment. It's the only gene therapy available for inherited retinal diseases. And so it was very important for us to determine whether or not they, in fact, had this um, particular gene as the cause. And the genetic testing came back a bit ambiguous. It wasn't clear cut. And so we were able to, with the help of a team from Sydney, actually, take some skin cells from both individuals, make stem cells, then turn those stem cells into retinal cells, and then, just like Anne does in the lab, test the function of that particular gene that we weren't sure about. And sure enough, after a lot of work, brilliant science work from the team in Sydney, we were able to show that, yes, that gene was the cause because it was proven in those cells in the dish to not be working properly. And so they've now both had gene therapy and their vision has improved. And so that is a great example of the power of stem cells in making a diagnosis. We've talked about
Mm.
lab work, we've talked about possible treatments in the future, but making a diagnosis is another key area. So that was an exciting use of stem cells.
Mm. So Matt has asked a question in the chat. Thank you, Matt. He says, thanks to the wonderful panel, wondering about the prospects of treatment for retinitis pigmentosa, for rod and cone cell restoration or replacement. Tom, do you have any thoughts to share with Matt on
Yeah,
that?
well, I get asked this question a lot by my patients, and unfortunately, for those reasons I sort of touched on briefly earlier, the complexity of the retina and the importance of making all those connections. We're not there yet. And then the surgical side of things is difficult too. I mean, we know we can sort of make these organoids, but how would you then get that into the back of the eye? How would you then have that layer connect to other layers? I mean, the most complicated connections is the innermost layer of the retina. The retina's bit back to front. The layer that detects light is actually the bottommost layer. The topmost layer sends out nerve fibers that go out of the eye, down the optic nerve, and then all the way into a very specific part of the brain where each fiber has to meet with another cell at a specific location to continue on that chain of the visual pathway all the way back to the visual cortex at the back of the head. So you can, I mean, it's just beyond sort of our ability at this stage to conceive of how you would put in a fresh layer of the retina and then have it make all those connections. I mean, it's just very, very complicated. That's why I mentioned earlier that perhaps a simpler layer which supports the retina, it's just a single layer of cells that sits underneath, like a set of tiles, doesn't have complex connections. That's probably a target that we'd get to first. But rod cone dystrophy is still a little ways off, I think. But a lot of people working on it.
So I guess I'm going to jump in, and I'm going to mention this later as well. But we do, so one of the things that we do in the lab is to grow healthy photoreceptor cells, so the light sensing cells that are dying in retinitis pigmentosa, and we can then transplant the cells back. So we replace these cells into blind mice. And I mean, I think we've been studying these procedures, the surgery, how to grow cells that are actually physiologically very similar to the photoreceptors. And we've studied the time points, what age makes these cells the best cells to actually get into the eye and then connect. As Tom was saying, it's such a complex environment, but the photoreceptors need to do two things. They need to connect with the layer. So it's what we call make synapses. So they connect, they hang it like touch each other and then send signals through that layer. And they also need to mature. They need to form this very specialized region that only photoreceptor cells have, which contain all the opsins. So these two things need to happen. So it is a complicated scenario, but we're working very much towards that. And in fact, a Japanese group has started clinical trials, and we had transplantation of 2 or 3 patients now with stem cell derived photoreceptors, not a purified population of photoreceptors. They actually cut the mini retinas that we generate in the lab, and they transplant that into the patients. We have a different approach. We purify the photoreceptors first, and we transplant 500,000 photoreceptor cells at the moment in animal models. We're going towards patients in the future.
I think that's promising. That's exciting that it is progressing on 2 different fronts. You don't all have to be pursuing the same pathways. Do you think, how far off do you think human trials are?
Well, look,
Large animal trials first, or
large animals first. That there is a handful of people that study it. So the Japanese are doing trials now. We have the group in London where I studied that. I presume in a couple of years we'll be starting clinical trials, definitely moving towards that. We have clinical trials with a slightly younger population of cells, also starting shortly in the US. And of course, I'm working very hard here in Australia to bring these therapies to patients here, because that is really my aim. We have a big grant where we are modifying everything that we do on the standard lab, right? In the lab that we just do research to now make these cells in a way that is compatible to go into human eye. So we need to do this good manufacturing practice production of cells so that they are good to go. And of course, we need to test these cells before they go into patients. We need to do more tests in animals and so on to make sure that the final product, it's good enough.
Well, you've got proof of concept though, so that's promising. Turning to you, Stephanie, you made news a few years ago with a treatment that used contact lenses, of all things, to transfer healthy limbal stem cells from a healthy eye in a patient to an eye that's been injured. Tell us about that research and where's that now? How's that work progressing?
Yeah, no, we did a clinical trial where we managed to treat a number of patients, and fortunately, we had reasonable success, about sort of 60 to 70% success rates. But we wanted to sort of optimize the procedure, so we've got additional funding, and we're using sort of devices that are able to increase stem cell numbers and quality. And looking at using sort of similar devices to transplant stem cells. So the stem cells on the front of the eye, as I mentioned before, are in the limbus, and it's very specialized. So we need a way to actually bring stem cells to the cornea, and then once they're there, protect them. And that's why a sort of a contact lens type device is good. This is one of the patients that we had treated in our initial trial who had stem cell deficiency following recurrent treatments for conjunctival melanoma. And there's a before and after shot there to show the success. But the technique that we developed was still reliant on taking donor tissue from the healthy fellow eye, and we were unable to treat bilateral disease. And so this is what we're working on and developing.
Yes. Is that something that you hope to see in the clinic as a something that people can ask for or be recommended?
No, that's definitely where we're headed, and we're sort of fortunate in treating the front of the eye that it's not as complex as the retina. If we can provide the correct cells, then they will make their own sort of connections. It's not neurological tissue, it's embryologically, it's sort of ectoderm, so more skin-like, and therefore easier to replace with stem cells.
I have a question here from Laurie S. that I think I'll direct to you, Tom. Laurie asks, I'm not an expert, but I think single mutation diseases may be amenable to CRISPR editing. Is that correct?
Yes, that is correct, and that's an approach that's been developed. There are different types of gene mutations. There are those that render the gene sort of inactive, in which case, the strategy there is just to try and bring a correct and working copy into the cell to fix that issue. But then there are scenarios where the change in the genetic code is the equivalent of throwing a spanner into the engine. It causes problems. It makes the gene go rogue or produce proteins that are damaging to the cell. So whilst you could put more of the right copy in, it wouldn't actually fix the damage that's being caused by the problems. And that's where CRISPR editing and other forms of gene editing are being developed to deal with that particular scenario. So yes, it's quite right, and the technology is being applied to emerging therapies for sure.
Mm. And thanks for that question, Laurie. And a reminder before I come to you, you can put your questions. It's over to the audience to ask their questions. Do put them in the Zoom chat. And if you want to direct your question to a specific panelist, then please do mention that. So, for example, Stephanie, can you tell us X, Y, and Z? Were you going to say something?
Yeah, so I was going to mention that we can use the organoids, so the mini retinas that we generate from stem cells from patients to test these gene therapies and the CRISPR editing. And the traditional way of doing it's with a viral vector. So it's a modified vector, a virus. that it's safe. It has been tested to be safe. And this viral vector, it's almost like a delivery truck. It goes into, it contacts the cells of, in this case, the organoids or the patients, and delivers the copy of the gene that we want to enhance the cell with. So if that gene is non-functional or is missing from that cell, that viral vector can deliver that gene, and hopefully that gene is then on, and we have a healthy protein at the end, and hopefully the cell then becomes healthy again. Now, with CRISPR and some of the diseases that we study in the dish, such as Stargardt or Usher, some of the very common diseases that affect the retina, these genes, the mutations on these genes, so these genes are too big, basically. So they're a bit more complex to treat because they actually don't fit inside this viral vector. So the truck cannot deliver these genes, even though if we were able to deliver, we would perhaps treat the cells. In this case, we can not do that because they're large. So that's when CRISPR going in with this specialized enzyme that can find the exact point in the genome where the correction needs to happen. So what we do with the gene therapy in this case is deliver this enzyme, and then the CRISPR mechanism can act on those mutations and then correct specifically, very specifically, the mutation, and that can be done for Stargardt's and Usher's and other genes as well.
It's interesting that you talk about the viral vectors as delivery vehicles that deliver the corrected gene code. Tom, when we were talking in preparation for this webinar, you mentioned that stem cells can also produce some things that are therapeutic for tissues, including eye tissues. Exomes, I think it was, that you referred to. Can you tell us a bit more about that? What's the potential there?
Yeah, well, stem cells have been used to produce tiny little vesicles, like little sort of tiny little membrane-bound capsules, if you like, that are released by the cell to deliver a therapeutic agent to a target cell. And so that's a potential property that can be harnessed and used to deliver therapies to the retina and other parts of the eye. And so that's an area that we're looking at the moment, because as Anna said, the viruses that are currently being used are quite restrictive in the payload they can deliver. And ABCA4 being such a common inherited disease is too big to fit in a viral vector. And so stem cells may provide an alternative delivery van, a bigger one, that could fix that problem.
Thanks for that, Tom. Look, I might direct a question to you, Stephanie, from Ellen. Have you come across Sjogren's disease? Is that how you pronounce it?
Sjogren's disease. So this is a dry eye disease where it's autoimmune based, where you have an attack on your lacrimal gland and also salivary glands, so you get dry eyes and a dry mouth. And look, unfortunately, at this stage, there's no regulated stem cell therapies for Sjogren's syndrome. It's important to remember, when you're looking into these treatments, because I know there's some treatments out there that haven't been shown to be effective in a scientific way. So it's important to consider, whether a treatment that you're looking at actually has proper scientific evidence to show that it works and also to show that it's safe. Any sort of treatment that is very new and what we consider experimental should be delivered in the setting of a clinical trial where you give, appropriate informed consent and you're aware of the risks and the benefits. So do be aware there are some sort of so-called stem cell treatments out there that may not be something that you should pursue.
So it's not the sort of thing to book your ticket to Mexico or Russia for the
No, they have
or
totally different standards of medicine and therapy regulation there. So, with our therapeutic goods administration, we have to show that a treatment works and also is safe before it becomes on the market. And that, involves a series of clinical trials to really show that efficacy.
yeah, because I guess something that people don't often think of is they assume that either nothing will happen or their eyesight or other aspect of health will be improved. There's a risk, any intervention has a risk as well that you might lose what good health you have or what current vision you do have. Is that correct?
Yes, look, there were some unfortunate cases reported from clinics in America where patients thought they were getting a stem cell therapy into their eye. They thought it was part of a clinical trial. And what actually happened is the patients had irreversible vision loss from hemorrhage inside the eye and damage to the retina. And so it's very important to consider, the quality of a treatment, particularly the scientific evidence.
Yeah.
And I
Paul
think
has asked, yes and no? Yes, sir?
that's why it's so important that talks like this are very important because we can educate and say that, as Tom mentioned before, the last thing you want is stem cells transplanted into your eye because stem cells are highly proliferative. They keep growing and growing, and you're going to end up with a tumor and other things growing in your eye. You want it to be a stem cell that has become a specialized cell, in this case, the photoreceptor cell or a retinal ganglion cell for glaucoma, for example, and then transplant those cells. And there's years and years of studies to establish the correct age of these cells, how they're going to connect to improve that connectivity and the transplantation and the procedure itself. In the case of Stephanie, for example, yes, she does transplant stem cells, but they are again those more how does it have a restrict fate, right, Stephanie? They know they'll become eye cells or limbo stem cells, so they're going to repopulate and they'll become just that cell population. There's less dangers, there's no dangers then.
Yes, they're more specialized. I think
Yes.
that's been a very important thing for the foundation in supporting, researchers and funding their research is that at the end of the day, it's all about evidence-based medicine and making sure people are using treatments that are proven treatments. And you can't have proven treatments without researchers like yourselves, doing that research and providing that, that evidence base that people can trust and feel secure in.
Yes, and
I'll
I'm
go
going to
to
add Professor Muncie's done some work on, stem cell tourism, and the dangers of stem cell tourism where people, go to other countries to get an unregulated stem cell treatment.
yes,
Yeah.
yes. I think she's in the audience tonight, so I'll say a little, yep. Hello, Megan. A question from Paul. He's asked stem cells for cone viability factor or RPE restoration. Would anyone like to speak to that? That's what is RPE restoration and do stem
Well,
cells have the potential for cone viability? Tom, I think that might be your one.
the RPE is the retinal pigment epithelium, and that's that single layer of tile sort of cells that sits underneath the retina, and is a more simple structure, and therefore more amenable, relatively speaking, to replacement because it doesn't really make connections as such. Well, it makes connections to each other, but that could be established outside of the eye and then put in underneath the retina. I mean that has actually been done with sort of varying sort of success. But definitely that is on the radar, and people are looking at doing that because that could be highly beneficial to people with macular degeneration, where that layer can really thin out and become very damaged, and that then causes secondary damage to the retina. And that would support the cones. And so if the primary pathology is in the RPE layer, in the area of the cones, the cone cells, they're the ones that are right in the bullseye center of the retina, then for sure, you might be able to support those cone cells and prevent them from dying out. So it's like you can think of it like a complicated ecosystem or a rainforest. These cells are all interdependent. So you lose one layer, it can have flow-on effects to other parts of the ecosystem.
I like that description of it. Yeah. Neil has a question, and then we'll have a couple of wrap-up questions because we're almost out of time. Neil has asked, I'll take the second part of his question. I'm wondering how Associate Professor Anai and their team deliver their photoreceptor stem cells in their current animal models.
Yeah, so I think what the difficulties of the surgery. So we do it under the microscope with a contact lens that makes everything very large for us to be able to deliver the cells, and we need to deliver the cells to the back of the eye, and it's right in between that retinal pigmented epithelium, so the RPE, the carpet of cells that Tom just mentioned, and the retina. So and we actually don't have a space there. There is these two cells, they're touching each other. They're not glued together, but they touch each other. And when we deliver the cells there or the gene therapy, a pocket will form because the cells are just touching each other. Now, of course, that delivery, it's what Tom was mentioning, getting right to that space that is not really space. It's complicated, and it's something that very few surgeons in the world can do. And we do it in the animal, very thin retina. And we practice it a lot, but perhaps Tom can mention a little bit more of the difficulties in actually delivering that to in the patients themselves.
Thanks for that clarification. Clear, Tom? Yeah.
Well, briefly, it's a very, very tiny, tiny needle, which can be passed through the retina without damaging it. And you've got to get the position of the needle just right, so you're in that space that and I had mentioned, which is not really a space until you sort of create the space. I tell patients a little bit like sort of putting a garden hose, through a hole in your carpet and filling up the space between the carpet and the floorboards. There is a space that could be there if you create it, and obviously you want to be very careful because the retina is delicate at the best of times, but if it's a retina that's been affected by an inherited retinal disease, and it's even more delicate, and there are fewer cells there, and those cells are more vulnerable to anything that's disrupting their local environment. So yeah.
Final question before my last couple of questions. Leighton Boyd from Retina Australia has asked, how can a person with an inherited retinal disease who does not know their genetic diagnosis assist with research?
Well, I could have a go at that because that's a problem that we hello Leighton, by the way. That's a problem that we face not infrequently. 30% of people who have genetic testing will have an inconclusive genetic diagnosis. So that's a large group of patients. Mm-hmm. And it bridges nicely to a group of therapies that we haven't really talked about too much tonight, which are these sort of gene agnostic approaches where the strategy may relate to a more general neuroprotective effect. And a good example of that is a study that has been conducted, a phase 2 study that's been conducted in Australia called the Nacuity study. It's a tablet that people take that is a strong antioxidant, and oxidative stress or damage to the retina has been implicated in the type of damage that happens in inherited retinal diseases. So if we can block that oxidative damage with a strong antioxidant, then that could be very promising. It wouldn't cure the disease, but it would put the brakes on it. And if that buys patients a bit more time, holds their vision for a bit longer until maybe something more definitive comes online, then that's a plus. So people who haven't yet got a clear-cut genetic diagnosis can 100% sort of think about these therapies or participate in these trials potentially that have a more gene agnostic approach. And that Nacuity study is just one example.
And potentially buy some time.
Yes.
Stephanie, thanks for the cautionary words that you were saying before about the need for proven treatments. Where can people go to get good advice?
Yeah, we did a patient information leaflet with Ransco and Stem Cells Australia that is available that has some simple questions you can ask any practitioner offering you a stem cell treatment. We also wrote a position statement at the time, and we've just updated this with a consensus statement summarising what sort of stem cell therapies are currently available in the clinic and what's coming to the clinic to keep people informed.
Yeah. And Jackie will post that in the group chat for people to have a look at themselves. Tom, as our other clinician that can give us practical eye health advice, do you have any tips on how to look after our precious eyes and eyesight?
Well, I'd probably link back to those categories I mentioned earlier. If you're a young, healthy person, dare I say it, male, wear protective eyewear when you're out in the garden and working on a building site, because young men are, the ones who get these nasty traumas by and large. If you're diabetic, you've got to look after your diabetes. I mean, it's horrendous. It's absolutely shocking how, what we see in our clinic in terms of poorly controlled diabetics with terrible, terrible eye disease. If you've got a family history of glaucoma or macular degeneration, get your eyes checked. I mean, not necessarily, to the extent that you're becoming sort of paranoid or anything, but if there's a family history, go get your eyes checked. And if you've got an inherited retinal disease, the best thing really is to participate in research. Sign up to a registry, get involved with your local research institution. There are centres in each state of Australia, and we all talk to each other. So if there's an opportunity to get involved in research, you'll know about it if you're on one of these registries, but you might not hear about it or be as directly involved if you're not sort of on the research team's radar by being on a registry.
It's now time for us to just about wrap up, but I did want to give each of the speakers an opportunity for a parting word of wisdom or thought for people to take home today. We'll start with you, and
I look, I think the therapies, development of therapies for retinal diseases has progressed so much in the last few years, in the last decade, that we can only have hope that, more and more therapies will be available. It is a long way. Research takes a long time to do proper research and, have the approvals and everything that we need to do to develop a new therapy. But I think there is a lot of good researchers out there working hard every day to bring these therapies, and the technology is improving. We have, who would think 10 years ago that we would have mini retinas growing in the dish where we could test therapies? It is progressing remarkably. So I think that there's very much hope for the future.
thank you. I'll go to you, Tom, and then finally Stephanie. So, Tom.
Well, I would just say it's a very exciting time to be involved in eye research, and it's an exciting and hopeful time to be a patient with an eye disease. And these things all happen or advance through small steps being made by scientists and with the participation and involvement of patients and research participants. They're all small steps, but the small steps are happening faster and faster, and we're really sort of, I think, on the edge of an exciting era of combination therapy, gene therapies, gene agnostic therapies, and potentially stem cell therapies. So there's a lot to be hopeful about.
Thank you, Tom. And finally, Stephanie.
Yes, look, stem cell therapies hold a lot of promise for restoring vision, but at the moment we have to also remember the reality of what they can actually provide.
Thanks so much. That's all we have time for. Ladies and gentlemen, thank you for your questions, and please join me in thanking our panelists, Stephanie Watson, Tom Edwards, and Anai Gonzalez Cordero. And there are some nice little, yes, the little hands emojis. We very much appreciate that. Now, if you or someone you know has questions about the stem cell treatments that are currently available, we do have an information resource to help. The Foundation Guidebook, What You Need to Know About Stem Cell Therapies, can be downloaded from our website at www.stemcellfoundation.net.au. And we have put that link to the Foundation website in the Zoom chat. While you're at the Stem Cell Foundation's website, it's nearly the end of financial year. So if you'd like to make a tax-deductible donation to the foundation, that would be just lovely. So that's at www.stemcellfoundation.net.au/donate. We use donated funds to provide public and patient education, such as events like tonight's webinar. Our next event will be an in-person event, we think, in Melbourne in November. And if you'd like to hear about events and stem cell news, then sign up for our newsletter via our website. And the vast bulk of the funds we raise do go to supporting promising Australian researchers. And as I mentioned in their introduction, Anai and Tom are just two of the dozens of researchers that the Foundation has been able to provide funding to thanks to the generosity of our donors. So we very much do appreciate the support. The scientists we've funded are working on everything from basic fundamental research to improving treatments for cancer, multiple sclerosis, cystic fibrosis, heart disease, diabetes, other forms of vision loss, osteoarthritis, lung cancer, and more. And you can read about them on our website. If you'd like more information about making a donation, please contact our General Manager, Graham Megan, via inquiries@stemcellfoundation.net.au. Thank you again for joining us tonight and have a great evening.